Speak Student!
Molecular genetics, electrophoresis.
A la Shmoop. Okay, we're on DNA technology with Dr. Ruth Tenen. So the DNA then has to be sort of separated or segmented and managed in various ways. Talk us through the process and basically the process of electrophoresis. How does all that kind of come together?
Yeah, so electrophoresis is a really useful tool, and it's basically a way of separating DNA by size using an electric field. So you add your DNA into an agarose gel, which kind of feels like Jell-O. If you zoom in really close on it, it kind of looks like a spider web, so it's all like tons of webbing all over. So what you do is you add an electric field that is negative where you put the DNA and positive at the other end. And the DNA, which is negatively charged, will kind of run down the gel toward the positive electrode.
It's like a magnet.
It's exactly like that, yep. And the smaller the piece of DNA, the faster it's going to be able to make it through that web. So it separates things by size.
And we've done this after we've chopped it up with restriction enzymes. And then we study the patterns, a little bit like if you collide atoms, you'll look at how they smash together and it'll give you characteristics of the medium it's in and so on.
Yep.
Is that it? We're sort of looking at the trail of corpse of, sort of what's happened in this collision? Or
so there's a bunch of different uses. So once you've got those pieces separated, one thing you can do is actually cut them out of the gel and use them for other purposes, but you could use them for forensics. So you could have a pattern of DNA from a suspect and a pattern of DNA from the scene, and you could just compare what they look like, what the fingerprints look like.
Wow, cool. So we cut them out of the spiderweb gel, and we want to use them for something else.
Yep.
Is this, I think I remember reading a Google article about, our being able to, we tried to grow a frog, and we didn't grow the whole frog, we grew an arm. Like, is it where you can cut them out and like grow cool stuff in
most cases, it's not super dramatic. So you might have a gene that controls some small thing that you could put into a new organism, and it could give it a new trait. So for example, like you could have a plant that normally is, not resistant to a certain insect, and then you add the special gene to it, and now it is.
What's the process of electrophoresis?